Good afternoon, everyone. I'm Fatima Zaria, Department of Veterinary Microbiology, Faculty of Veterinary Medicine, Ahmadu Bello University, Zaria, Nigeria. I'm here to make a presentation on the assessment of antibacterial effects of Terminalia avicennioides on selected local and reference strains of gram-positive and gram-negative bacteria. Bacteria are tiny organisms that cannot be seen with the naked eyes, and they are found almost everywhere on Earth. Generally, bacteria are divided into gram-positive and gram-negative bacteria. Examples of gram-positive bacteria include Bacillus, Staphylococcus, and Streptococcus, while examples of gram-negative bacteria include Klebsiella, E. coli, and Pseudomonas. Reference strains are strains that have been widely characterized and they are used for quality control studies, while local strains are strains that are obtained from specific geographical location and they represent regional resistance pattern. Often, antibiotics are used in management of disease condition caused by these bacteria. However, overuse of antibiotics has led to the global antimicrobial resistance crisis. The rapid emergence and spread of resistance bacteria like Methicillin-resistant Staphylococcus aureus and other bacterial species has rendered antibiotics ineffective, thus complicating infectious disease control. In response to antimicrobial resistance, there is renewed interest in plant-derived antibiotics, which are natural substances with antimicrobial properties that are found in plants and are used as alternative to conventional antibiotics. Materials and method. The study was conducted at the Antimicrobial Resistant Sentinel Laboratory in the Department of Veterinary Microbiology, Faculty of Veterinary Medicine, Ahmadu Bello University, Zaria, Nigeria, and it was an experimental laboratory-based design. Source of the bacterial isolates. All the reference strains were sourced from Noguchi Memorial Research Institute in the University of Ghana, and all the local isolates were sourced from Ahmadu Bello University, Zaria. The stem bark of the plant extract was collected from Samaru campus in Ahmadu Bello University, and it was taken to the herbarium of the Department of Biological Science for authentication. It was then washed, dried, and pounded into powdered form, and 0.4 gram was dissolved in 2 ml of water, which was the stock solution, and serial dilution was made across 6 graded concentrations starting from 100 milligram per ml. The inoculum were prepared based on standard. The antimicrobial susceptibility test was also done based on standard. The agar-well diffusion method for determination of minimum inhibition concentration was done based on standard. Results and discussion. We subjected all the bacterial isolates to colony morphology, and these are the results. This is Methicillin-resistant Staphylococcus aureus on mannitol salt agar. This is Staphylococcus aureus on blood agar. This is Bacillus on blood agar. This is E. coli on eosinophilic blue. This is Klebsiella on McConkey agar, and this is Pseudomonas on nutrient agar. We also did gram staining results. These are the results for gram-negative bacteria. They appear as gram-negative bacilli arranged in pairs, and this is Staphylococcus aureus, which appears as gram-positive cocci arranged in clusters. All the bacterial isolates were subjected to standard biochemical tests, and this is biochemical test that was conducted on E. coli. The antimicrobial susceptibility profile of the gram-positive bacteria, methicillin-resistant Staphylococcus aureus was resistant to 4 classes of antibiotics that were used in these studies, which we confirmed it to be a multi-drug resistance organism. Bacillus was resistant to septrin and oxacillin. These are pictures showing the zone of inhibition of the bacterial isolates. For the gram-negative bacteria, E. coli was resistant to amoxicillin and cefoxitin, while Klebsiella and Pseudomonas were resistant to at least 3 different types of antibiotics that were used in this research, which made them to be a multi-drug resistance organism. The antibacterial activities of Terminalia avicennioides were relatively stronger across higher concentration. And the minimal inhibition concentration for gram-negative bacteria is 6.25 milligram per ml, and for gram-positive bacteria is 3.125 milligram per ml. In a study conducted by Ajayi et al. 2020, they showed the leaf extract of Terminalia avicennioides. The minimal inhibition was for Bacillus subtilis was within 2.5 to 5 milligram per ml, which is in contrast to this study. As we can see, Bacillus here is within the upper range, and we attributed it to species variation. Also, Staphylococcus aureus and Methicillin-resistant Staphylococcus aureus shows good antibacterial activities against Terminalia avicennioides, which is in correlation to a study conducted by Aliu Amo and Isa et al. 2023, who showed the methanolic root extract of Terminalia avicennioides has good antibacterial activities against Staphylococcus aureus. In a study conducted by Mann et al. 2019, they reported that plants that are high in tannins, glycosides, and flavonoids have good antibacterial activities against E. coli, Klebsiella pneumoniae, and Pseudomonas species, which is in contrast to this study because we only made use of one plant extract, which is Terminalia avicennioides. And Terminalia avicennioides is only high in tannin. It has moderate glycosides and flavonoids. In conclusion, all the bacterial isolates were phenotypically confirmed, and the minimal inhibition concentration for gram-positive bacteria was 3.125 milligram per ml. Gram-negative bacteria was 6.25 milligram per ml. Also, methicillin-resistant Staphylococcus aureus, Klebsiella pneumoniae, and Pseudomonas species were confirmed to be multi-drug resistant organisms. Acknowledgement. We authors, we acknowledge the Department of Veterinary Microbiology, Faculty of Veterinary Medicine, Ahmadu Bello University, Zaria, Nigeria, for their cooperation during the course of carrying out this research. Thank you very much.